Okay, so when we're talking about DNA replication, we're talking about the concept of taking a DNA molecule, and a single DNA molecule, double helix, is a chromosome, right? Not all DNA molecules are chromosomes. There are, for instance, fragments of chromosomes, but all chromosomes are DNA molecules. Is that clear?
Yeah.
So you have a DNA molecule, when it's all wound up, it forms these things called chromosomes. The only time you wind them up is when you're ready to send them somewhere. The only way you can send them somewhere is if you made copies of them. And so you have two copies. You have two copies of DNA for each one of your chromosomes. You have 23 pairs. One came from mom, the yellow one came, let's say the yellow one came from mom and the white one came from dad. And I like the yellow and the white because they show up well on the green background. So you have the yellow, and so you have two, each of these has the same information, same genes. They're slightly different, right? The sequences are slightly different, but because of mutations. Is that clear? Mom's DNA is a little different from dad's on each one of the chromosomes. Even the ones that should be identical, both of these are chromosome number 1. That's called genetic variation, right? So we have these differences in these gene sequences on each of these chromosomes. One came from dad, one came from mom. If you're going to take this cell and you're going to make 2 copies of this cell, you need to make copies of the DNA before you do that, right? Otherwise, you can't have 2/3 of the DNA going into one and 1/3 of the DNA going to the other cell. The cell won't work well. You can't have too much or too little. You have to have exactly 23 pairs. Why are there pairs? Because for all 23 chromosomes, one chromosome came from dad and the other one came from mom. Is that clear? The packaging of the DNA leads you to have, once you do the copying, once you copy these, you copy the yellow and you copy the white, what you end up with is a chromosome all wound up, and you've seen these drawings in the book if you've read your book, as you should have by now. It's been a week since you got your assignments. It's been a week today. You got your assignments last Monday. How may I help you? So we copy each of these chromosomes, and so you have here, you see that there's a copy here of dad's chromosome, right? You notice that there's 2 copies, right? And they're stuck together. That's what you see when you look under a microscope and you see chromosomes. You see them like this, because the only way you see them is when they stop being chromatin and become what we call a chromosome. In other words, a DNA molecule all wound up around histones. And the same thing happens in mom's chromosome. It gets all wound up and duplicated, so you have copies, and they're held together by a centromere in the center here. Also another protein, and that is mom's chromosome. So you have 2, but how are we going to get that? You're going to separate them so that you get 1 copy of mom's chromosome in one cell and 1 copy of dad's chromosome in the other cell, and 1 copy of mom's chromosome in one cell, or dad's in one cell and mom's in the other cell. So 1 of each in each new cell. Now you do this 23 times, right? Because there's 23 chromosomes. Are we understanding what I'm saying?
Yeah.
So you have a copy of all the genes, all the information, all the genetic information from mom and from dad in each one of the cells. Every cell in your body is genetically identical. They all have the same genetic sequence, except your sex cells, what we call gametes. Except your sex cells that we call gametes. Your gametes are a little different, because you run into a problem if you do this, if you keep making copies of the DNA and you keep dividing. That's fine. That can happen, right? You divide, you double the DNA, you divide it. So you start off with 23 pairs, you double it, right? So let's think about the math there. You start off with 23 pairs, which is 46, right? Right? 46 chromosomes. Doubles for each one of the 23, right? That's 23 pairs. Do you hear what I'm saying to you? 23 pairs. Pairs means 2, right? 2 times 23 is 46. So you make copies of all that. So after you make copies, what do? How many do you have? How many do you have? 92. So now you have 92 chromosomes. So that you can do what with those 92? Split them up. Why do we want to split them up? Well, that's true. But so you can make 2 cells, each one having what? The same number and the same DNA. That's correct. That's perfect. That's exactly right. So you can have the same number and the same DNA in each cell. Is that clear to everybody? So that makes sense. That's why we double the DNA. That's why you have to make copies. So you have every cell in your body has exactly the same DNA. Except for, this process, what you see here, is called mitosis. And some of you have already studied it in K through 8. You should have studied it already. You should know it. Should have learned it. If you didn't, you're going to have a winter package that's going to go over it. Yeah, that's why you're going to go over it. Because if you have seen it, you haven't seen it since 8th grade. If you have seen it, then if you haven't seen it, you need to see it. So 46 in each. Does that make sense? Or again, I want to say again, 23 pairs. 23 pairs. Is everybody okay with that? Yes. So that's mitosis. That's how you went from one single cell. Your mom and dad got together, had a nice evening. Then they made you, right? There was one single cell, and then you did this a bunch of times, and you became you, 1 trillion cells. That's a big change, right? And then it make copy after copy after copy. Now with meiosis, or meiosis, however you want to pronounce it, we're going to do something different. And why do we have to do something different? Because imagine for a second, when your mom and dad, your mom's cells had, we said 46, right? 23 pairs. Dad also had 46 pairs. 23 pairs. When they got together and they made you, how many DNA, how many chromosomes would you have? 92. Well, that's no good. You're supposed to have 23 pairs. You're supposed to have 46, not 92. And what happens when you get together with some other kid, well, hopefully adult, and hopefully married, and a long time from now, after you have jobs, you can afford a baby. So you get together, you make a baby. Your baby would have what? 184. Well, this is not sustainable, is it? This doesn't make any sense. So we can't do it that way. We have to do something. What do we have to do first? In order to make this work, what do we have to do? Divide. We got to divide. We got to get rid of some stuff, don't we? Meiosis is doing mitosis, but doing mitosis with one big change, and that's that making that division first.
I was thinking about the plants.
Yeah, go ahead. What?
That's like in 8th grade we learned about how the plants, like one was asexual and then the other one.
Oh, okay. Yeah, that's slightly different. Yeah, that's slightly different, but still related, but slightly different. All right. So with meiosis, what we have to do is we have to take in men, in let's say the males, we're going to do something called spermatogenesis. Who knows what that is? In females, we're going to do something called oogenesis. Oogenesis. I don't know what filthy thing someone said, but I'm not ignoring it. All right, so oogenesis and spermatogenesis, all right? Ooh is same root word. It's a Greek root meaning egg. All right? Ova, ovary, right? So oogenesis is the is the making of what?
Egg.
Of the egg. And spermatogenesis is the creation of the sperm. So to make either one, both of these depend on meiosis. So the first thing we're going to do in both of these, and I'm not going to talk about the different changes. There's a lot of other changes that happen. Yeah. I'm not. So oogenesis and spermatogenesis, you're taking those 46 chromosomes, and the first thing you have to do is, and I'm going to go ahead and draw it out instead of putting the letters in. Can I use an example of one chromosome like we did above? Yeah. And I'm going to draw a cell with one chromosome from mom and one from dad. And the first thing we do is make copies. So you have to make copies. You have to make of the DNA. So we only have one of these. So that's the first step. And when you make copies, you have 2 of each chromosome, right? Hence, copies. Then, just like in mitosis, just like above, right? Just like we saw up there, we're going to take and we're going to split them. So each of these is going to split. In mitosis, it splits. In meiosis, it does too.
So in meiosis, it does one?
No, meiosis, you're going to do it, actually, you're going to do it twice, not once. So in meiosis, we're looking at spermatogenesis, and what we're going to do is we're going to take this, and we're going to take mom's homologs, or that's what we call the chromosomes, and we're going to take, they're going to move, and dads are going to move together. So they're going to move like that. See how they're still stuck together? So they're still, do you not see them together? Do you not see 2 chromosomes? Listen. If you don't see it, ask. Don't sit there and play games, because you won't be playing games when you don't get a good grade on your test, right? So why are you playing games now during the class? So if you don't see something, you don't actually see it, it's probably because you didn't read your homework. But whatever the reason, raise your hand and ask a question. So here is a chromosome. You see there's only one line. You make copies of it. I drew it up here. You see it drawn up here? This is mom's and this is dad's. We make copies. You see how it forms this shape? Do you see that shape? Yes or no?
Yes.
So here is that single line that would be analogous to this one. You make copies. It takes on this shape. Do you see that?
Yes.
These 2 move together to this side. These 2 move together to this side. We call each of these homologs. They have the same DNA.
Homolog?
It doesn't matter. It doesn't matter. So now, of course, we're trying to get half the DNA. Right now, we had what did we have up here? 23 pairs, right? Here we have what? 46 pairs, or 92, right?
Yes.
Now we're back to 23 pairs. That still doesn't help us, because if we take these cells at this stage and combine them with the opposite sex cell, right? You're going to have 46 again. 46 pairs or 92. We don't want 92. We want 23 pairs. So what do we have to do now? Yeah.
So when you divide each one by,
Both of you are right. Yes.
take one part off and put it on the other one.
That's both of you are right. So we do it again. We divide again. We divide it, right? Just like you say. We divide again. We divide into 2. Into 2 cells. Each. We divide into 2 cells each. So the first one, and we'll talk more about what the advantage is. How may I help you? I have no water other than what comes out of the faucet. There's some cups in there. So the chromosomes are split up into cells. So one copy of each chromosome goes into each of the 4 cells. So now instead of 23 pairs, well, how many do you have? 11. Just 23, right? Instead of having 2 number 1s and 2 number 2s and 2 number 3s, you have 1 of each. Does that make sense? And so we have just a number 23. Not 23 pairs, but 23 single copies
So like it's 23 of all of them? I mean, like,
in each one. That's right. There's 23 and 23 and 23. That's why they call that place that sequences your DNA, what do they call it? 23andMe. Why do they call it 23andMe? Because there's 23 pairs of chromosomes in your DNA. So 23 single copies in each of the sets. So this would be 23 copies of the genes, of your chromosomes rather, not genes, but 23 copies of the chromosomes in each of your eggs, in each of your sperm. So if one sperm meets up with one egg, they combine their DNA to get what? 46, or 23 pairs. You go from 23 singles to 23 pairs. So you have chromosome number 1, 2, 3, 4, 5, all the way to 22. And then you have 2 X's. This gentleman has chromosome number 1, 2, 3, 4, all the way to 22, and an X and a Y. He puts half that DNA in one sperm. You put half your DNA in one egg. When you take, if I took your sperm and your egg and I combine them, you get 46, or 23 pairs.
so 23 and 46 is the same thing? Yeah.
The number 4, what's 23 pairs? 23 times 2, which is 46. Right? But here we have 46 what? 46 pairs. So here we actually have 92. Do you see that? Because 46 times,
But how does that change?
we split it in half. You're splitting. The whole point is that you want to, you're going to divide everything. If you have to make copies before you divide, or you're going to have a different number every time you divide.
So you say,
Go ahead.
so in the real, like, with the example that you were just saying with the 2 people, is making a copy having a child?
Yes. Well, no, making copies is getting ready to have a child. The combination, combining your copies, these, let me label this so everybody is sure what we're talking about here. Sorry, I always hate unfilled triangles for some reason. It weirds me out. Must be OCD or something. These are called, where? Oh, Jesus. All right, so these are called gametes. Gametes or sex cells. They're the only cells in your body that are different from the others. Every other cell in your body has exactly the same DNA. The gametes have a different combination. They've been recombined, and they've also been separated. They have half the DNA, and they don't have the same combination of DNA as the rest of your cells.
Is this just for the males or just for both?
For both. That's a good question. Oh, here we go. They're going to announce what I already told you.
Just a reminder that all after-school activities are canceled for today. Thank you, and you need to exit the building. Thank you.
All right, can we focus? Because we don't have a lot of time left. There's only like 10 minutes left in class. And let any question be pertinent to the concept. Yeah?
You going to go early college?
I'm going to have to say no. Because it's 3:06, and by the time you get down there, come back up, the bell's going to ring. You know what I'm saying? You have to go all the way to the basement. If it's an emergency, go ahead. But I'm leaving at 3:18. If your stuff's locked up, see the principal. All right? Yeah.
You can go to A&D, that bathroom working.
I don't know. I wouldn't do. They told me the bathroom wasn't working. I don't know what's going on with that. You take your chances. Something happens. You know, something gets thrown up on you or something that's on you. Please don't take your stuff with you. You're gonna get in trouble. Just run, and if I leave before you get back, see somebody has a key. Get in here. Yeah.
So, meiosis only happens in sex cells?
I'm sorry, I can't hear you because somebody's talking up front. Go ahead.
Meiosis only happens in sex cells?
Yes, meiosis only happens in sex cells is correct.
So then mitosis happens.
Everywhere else. All your other cells. When you cut yourself, mitosis is happening. Mitosis is division of the nucleus. Meiosis division. Cell Replication is copying everything. Mitosis and meiosis are only part of the cell cycle. Yeah, we'll cover the cell cycle later, but go ahead.
Is mitosis kind of going to asexual reproduction kind of?
Yes, assuming it's a eukaryotic cell, yeah, because you have to be a eukaryote to have mitosis, right? Has to be you have to have a nucleus. So bacteria don't do mitosis; they do binary fission. They still do exact copies of the DNA, but they do it through differently. All right. Yeah.
I'm thinking of it how we did in 8th grade and with the plant food. That's how we learned.
Yeah, some plants do asexual division. That would be mitosis. That's correct. That idea is correct. Meiosis is sexual reproduction. That's when we recombine our DNA. There's an advantage to that. You have a larger variety, and therefore your population is more capable of surviving changes. The more alike we are, the less likely we are going to survive a big change, like an ice age, or a meteor hitting, or food supply change. The more diverse we are as a people, the stronger we are as a population. That's the reality. Yeah.
Do you know what? When you do meiosis, how come is it the same process? Because it's confusing me. Is it like new strand and old strand and sequence versus replication and conservative replication?
Well, we're going to talk about that. All that is talking about how do we make a copy. That's not mitosis. That's not meiosis. This is talking about how we separate them. Are we clear? The question that we're trying to answer in this past 2 or 3 days, over the weekend you should have read about it, is how do we make copies of the DNA? How do we get to this point during synthesis, which is a part of the cell cycle that looks at copying. So now let's go ahead and take this and look at how do we make a copy. So again, everybody should have read the chapter. The chapter discusses this in detail. This actual picture, and I think a better one, is actually in the chapter reading. It's part of your homework assignments. One of your homework assignments as you look at this as well. Out of all this that you drew, you have this double helix, right? They're wound up, they're tangled. Do you guys have earbuds, right? Yeah. For your phones, and you put them in your pocket or your purse. Most of the time, they're all tangled up, right? What would happen if you took each of the earbuds and you pulled on them just to separate them? They would turn into a knot, right? They would get all tangled up because the more you pull on one end, the other side gets all tangled up. Well, in order to read the DNA, your cell has to separate the two strands. If it started to separate them like this and pulled them apart, you'd get a knot up here, right? Well, there's this enzyme. You do not have to know this enzyme. So when I tell you you don't have to know it, that means don't worry about it. You can erase this or scratch it out if you used a pen. I don't care. You have this enzyme called topoisomerase gyrase. And all it does is, as you pull the two apart, it untangles the other end. Is that clear? Mm-hmm. That's all you need to know about that. You don't need to know that. I just want you to let you know that that's how it works. Now, something you do need to know, and it's in your reading, when you separate, these two DNA strands are sticky. The A sticks to the T's, the G's stick to the C's, right? They're very sticky. Hydrogen bonding is a sticky business, right? So they're separated. If you don't, you need something to help stop it from sticking back together again, right? So we separate them. We got to keep them separated long enough to be able to read and make a second copy. So what we do is what you do when you're remember how we did those paper projects? You had tape, these long strips of tape. How do people? I saw people do this. I saw people do it the wrong way. I saw people do it the right way. When people try to do it the wrong way, they try to leave here with these long strips of tape and go back to their table. What happened 9 times out of 10 before they got to their table? It touched itself or something else. It's all stuck to each other, stuck to paper. It's just a mess, right? So the people that were a little smarter, they took the two ends of the tape and kind of stuck it to just the ends to each other, or they wrapped it around their finger. So they stuck the tape to something else, so it wouldn't stick to itself. Does that make sense? So So that's what your body does. So these stick together, right? So what they do is you have these single-stranded binding proteins, SSBPs. They attach to the DNA. And what does that do? Stop it from sticking to itself.
So them little things, they stop it from sticking.
That's right. So that makes sense, doesn't it? All right. So the enzyme that makes the new DNA copy, this is the old one, right? You see it here? And this is the new one. Does everybody agree? Does everybody see it? All right. So DNA polymerase means it's making a polymer of what? DNA. DNA polymerase makes a polymer of DNA. DNA polymerase, you take one nucleotide and attach it to the next, attach it to the next, attach it to the next. And you keep putting them together, right? That's what this is. That's what this red line is. That's what DNA is. These two strands are complementary. They are not identical, the new and the old. They're not identical. They complement each other. I don't know why this is this way. Hold on. Okay, so when we're talking about the old and the new strand, what you have to understand is if you have A, T, G, C, ... for a million times over, different sequences for different people. What do you have on the new strand? T, oh, let me do a different color. T, A, C, G, ... right? Are those identical? No, they are not. But they match, right? There's a difference between matching and being identical. And over here, the old strand, on the other old strand, so this is one side, right? This is up here, right? What's down here on this other side? This is what? What is, this is the new one. You see that's new? Do you understand that?
Yeah.
Well, what's the old one on the other side? That's right, TACG, good. Because that's the matching, that was the complementary strand of this one, right? This one and this one complement each other. When you make the new strand on this side, what are you going to make? ATGC, ... Now, what I want to point out is that even though you did not make an identical second strand, right? The new strand is not identical to the old one. The new and old are not identical. In the end, what do you end up with? Isn't this identical to this?
You said it wasn't.
So you're not listening. I said, even though the old strand is not identical to the new one, when you're done, you will have two chromosomes, two double-stranded DNA molecules that are identical. Do you agree? Yeah.
Where's the neutron come from?
What did you say?
Where's the neutron come from?
What neutron? Oh, the new strand is made by DNA polymerase. DNA polymerase is making the new strands. That's what we're trying to get to. That's what we're trying to understand. So let's look at the new strand. Let's talk about where it comes from. So it's unzipped by helicase. There's this enzyme called helicase. There it is right here. Helicase opens up the helix. That makes sense, right?
Yeah.
Then DNA polymerase starts to read it, but only from 3' to 5. It only reads from 3 to 5. It does not read from 5 to 3. That's a problem for the DNA, for making copies of DNA, because if you're going to make copies on the other side, if this is 3' of the original strand, what is on the other side? What's this on the original strand? 5. So if this is 3, this is 5. DNA polymerase only reads from 3 to 5. There's no open 3' end on this end. So the only way to solve it is to make a free 3' end, and we'll continue this evening in the video I will send you. Good luck. That's this video completed. I'll see you tomorrow. We're going to get into translation tomorrow, so be ready. All right, so that's great for DNA replication, how do we make copies, and it goes into mitosis, and according to the state, we're not supposed to cover mitosis. That should be covered in 8th grade, but we're going to touch on it. I'm going to give you a winter break packet on it because I think it's important that you review it. You should know it, you should review it, mitosis, what is mitosis, how does it work? You should have, I don't know if they're gonna ask you questions on the end of course exam, they shouldn't because they say it's not in the curriculum, but who knows? Meiosis is something you need to know about, and without mitosis, you won't understand meiosis. Although again, the state says you should know what meiosis is.
Is that the same as Miles?
Meiosis, meiosis, yeah, and meiosis if you'd rather. Potato, potato. All right. Tomato,
I'll be
tomato.
having the same words, even
Do you eat tomatoes? Do you have your aunt versus aunt? I don't have aunts.
My auntie.
I don't have an auntie. I have an aunt. I know. I've heard it all. Please, let's move on. All right, so that's great for DNA replication. Now, there's a thing. What is DNA for? How does this DNA in the nucleus, this is the nucleus obviously, how does it control what goes on in the cell? We've been talking for a very long time about these things called proteins. And we've been talking for a very long time about these organelles called ribosomes. There's a big and a small one. DNA never leaves the nucleus. In mitosis, the nucleus disappears and is reformed. 2 nuclei are reformed after mitosis. That's part of mitosis. But the DNA never leaves the nucleus. The question is, how do you get the information? DNA has information, we all agree on that, right?
Yes.
It's your book of life. How do you get this information from here in the nucleus to the endoplasmic reticulum that has the ribosomes, the rough, right? Or the ribosomes, the free-floating ribosomes in the cytoplasm? How do you get the message out of the nucleus to the cytoplasm? Well, tomorrow you're going to have an activity that's going to record. You're going to work alone on those activities, and you're going to have to use what's called the universal code to transcribe and then translate a protein. So there's 2 steps. There's 2 steps. Again, read your chapter. We're in section 3 now. We're done with DNA replication. We're moving on to transcription. You should be done with chapter 12. I know some of you aren't. I'm really feeling bad for you. All right, so transcription. The answer to your question is, how do you get the information out? You use RNA. That's how eukaryotic cells get the message out. There are many forms of RNA, In fact, a ribosome, let's go ahead and make a list of the different kinds of RNA. There's ribosomal RNA, rRNA. The ribosomes are made of RNA. They're not made of protein. Most of the enzymes in your body are made of protein. It turns out that this enzyme, ribosomal RNA, ribosomes, are an enzyme, but they're made of RNA. And there's a big and a small. 2 small parts. One big and one small. rRNA, ribosomal RNA. There's another type of RNA that you should be familiar with, you need to memorize, and that's messenger RNA. mRNA. That's right, mRNA, messenger RNA. What do you think the messenger RNA is doing? It's taking the message. From what?
The nucleus to the ribosome.
That's right, you got it. It's taking the message from the DNA to the ribosome. So from the nucleus out. The single-stranded RNA can get out through the nuclear pores. The double-stranded DNA cannot.
But is the RNA in the nucleus or it comes?
The RNA is in the nucleus. In fact, remember we have the nucleolus. What does the nucleolus have? RNA. Remember the nucleolus is in the center of the nucleus.
So how is the RNA getting out of the nucleus?
Right through the nuclear pores. Remember those little holes?
Oh, yeah.
Remember I showed you in that game that we played?
Uh-huh.
Again, you're more than welcome to come and play it when there's time. All right. I wish you could all get it on it. I wish I'd give every one of you an iPad and put a bunch of games on it and books, and you guys could just do your work happily. But they do in other schools, but we don't do it here, and it's unfortunate, but we're moving on. I've suggested it many years in a row. I've suggested that we should give all our students an iPad when they get in the building. It doesn't have to be a big old iPad Pro, and I think an iPad because you can get apps, a lot of free apps, and they work, and there's very few viruses. And I think that they should allow us to just take you from beginning to end. These Chromebooks are okay. The good thing about Chromebooks is nobody wants them, so they're not stolen very often. The iPads, there's gonna be a lot of stealing and finagling. And people are like, they're $500. Yeah, but the books that we give you are $200, you know? We give you $200 books. Those algebra books are probably $200, close to it, new. And we give you 5 of them. That's $1,000 per kid. Why would we not want to just give you an iPad and then just put the content on it ourselves? That doesn't make sense. Anyways, we're wasting time. But that's a district level stuff. That's a conversation somebody else can have. So there are things that are beyond our control. So messenger RNA, rRNA, right? What's another one? Anybody know the third one? That I heard it. Yeah, tRNA. That's right. It's called transfer RNA. Good. Some people have read. I'm glad to hear it. Transfer RNA, and that is that these guys bring the amino acid. See, this brings the message. This brings the amino acid to the ribosome. Because remember, what's the purpose of the ribosome? Is to make a polymer of amino acids. So you need to know the 3 different types of RNA because they're the tools by which everything else is made. The DNA at this point is the messenger. So when they tell, you see why it bothers me that they only teach you in elementary school that the DNA, the nucleus is the control center of the cell. Right. That doesn't tell you much, right? It doesn't say much. It's just a very easy thing to remember, but it's not really saying much. All right, so transcription, the first step is to make the RNA transcript. And so you have this DNA, and it starts off like this, and then it has to open up again, doesn't it? You can't get to the message until it opens up. So what enzyme do you think is going to be making a copy of an mRNA? It's going to be making an mRNA. What do you think we're going to call that enzyme? What are we going to call an enzyme that makes RNA? Well, let's break it down. RNA for sure, right? RNA something. We know it's going to end with ACE. Polymerase, yeah. RNA polymerase. That's the enzyme that makes RNA. Polymerase, right? It's one word. I just hyphenated it to emphasize the idea that the word makes sense. The word makes sense. It's RNA polymerase. So RNA polymerase, what it does is it lands on the DNA just like DNA polymerase did, right? And it makes what? RNA. It makes an RNA. A long message starts RNA. Is that clear to everybody? Any questions at this point? So did anybody read anything in the book?
I have a question.
Yeah.
I find it. I'll put it down.
Do we need to memorize that?
Everything that's up on the board, everything in this movie that I say, unless I specifically tell you don't have to know it. The only reason I'm bringing it up is because you have to know. Do we have that question yet?
Oh, yeah.
Are we ready?
Oh, I have a question.
Yeah.
Okay, so in the reading, I didn't understand it. Well, I think you said something about it. It says the only way DNA varies among organisms is the the sequence of bases. What do they mean, the sequence of bases?
All right, the sequence, when they say bases, what bases are they talking about? The ATP, the nitrogenous bases. So humans might have A, T, G, C, A, T, but a dove might have T, T, G, A, T in the same gene sequence. Does that make sense? In other words, the difference between organisms is that we all have different genes. The difference between species, right?
Yeah.
Although we share a lot of the same genes too, okay? So even though we share a lot of the same genes, hence the idea of evolution, right? We also have many genes that are different, which is also part of the evolutionary theory, which evolutionary theory explains that. Why is it that we can have the same genes as a tree, but also have this different genes from a tree? How can both be true? If you're laying your head down, I'm making a note of it. You're sleeping in class, I'm making a note of it. I'm not going to make a big deal about it because I'm recording it, but I will be bringing that up during a parent conference.
So is the sequence the codes?
Huh?
Is the
It's the
sequence
code.
the code?
The code.
Oh, okay.
The message is the sequence. The order of the nitrogenous bases is the code.
So you see how I say it activates it and then it inactivates it. What's the point of activating it in the first place?
They're talking about the lac operon. That's an example. We can't get to an example until we'll talk about it. You want to leave this up here?
Yeah.
And we'll talk. Lac operon it's a classic example of how we can turn genes on and off. We turn genes on and off all day long. Every one of us has different genes on and off depending on who you are, and we'll talk about that in just a minute if we have time. Doesn't look like we will though, because it's 9:50 already. It's just the frustrating part when we have side conversations. So any RNA, I'm as guilty as you. I'm just saying, it's frustrating. RNA polymerase, making RNA. So what's the difference between making RNA and making a DNA? What are we going to do? If you have, come on guys, somebody actually say something that makes sense. If you have A, T, G, C, what are you going to make down here? A is going to make what on the RNA? No, it's not.
Huh?
U.
On the RNA?
U. That's the difference. T is going to make what? T is going to make what? A. A goes with T. G makes what? C. C makes a
G.
And so if there's another A, T, T, A, what's it going to happen now? Quickly. U, A, U. You see? Oh, jeez. U. Do you see how it goes?
Yeah.
That's really the main big difference between the two, RNA and DNA. Of course, RNA is single-stranded, DNA is double-stranded. Yes, of course, the sugar on the DNA is ribose, and on DNA is deoxyribose, right? Those are the three main differences. All right, so that's transcription. That's in short, but there's obviously a lot of steps. So you have this DNA molecule, DNA, I'm going to label it DNA. Is the whole thing this message? The answer is no. There's only a small piece that's a message. These pieces are called what? Not fragments. This is called a gene. That's what a gene is. How does the RNA polymerase know that this is a gene? How does it know that this is the code here, but not this over here? It's all one long DNA, it's all one long sequence, right? Why is this the hair color gene and not this part over here? Right? Why is this part of the DNA not being expressed? We call it expression, by the way. Let's make sure we're clear on that. Gene expression
Because it's a different code?
means you make a protein. Not all of the DNA makes protein. In fact, most of the DNA doesn't make a protein. There is no gene expression. Most of the DNA is not genes. So the question is, how does an RNA polymerase know to land here and start making the RNA, right? How does it know that this is the start? How? Well, the way it knows, like how does it know? The way it knows it is that this area here, right before the gene, is called the promoter.
So it promotes it.
It promotes it. It says, hey, this is the spot. Right after me, there's a gene. Start doing it. It's chemically, obviously it's not actually calling out to each other, but it's a chemical reaction. There's an area of a promoter, there's an area of the DNA right before the gene that says, this is where you attach RNA polymerase, and the RNA polymerase attaches. All right? And as soon as it attaches, it starts to read it just like DNA polymerase did. But of course, it makes an RNA and not a DNA. Well, how does it know when to stop? How does it know? How does it know? There is another reaction. How does it know when to stop reading? It starts. There's a start, and we call this the area of the promoter region, right? So what do you think we call the end? Just anybody? It's called termination. So you have the promoter and then you have a termination. So the two sides of the gene are bookended, right? One end you have a promoter side, and the other end is a termination sequence. So there's a sequence of DNA that says, that's it, we're done, and the RNA polymerase falls off. So then you end up with a protein, right? Not a protein, excuse me. You end up with an mRNA that's exactly as long as the gene. You see that? This is complementary, except it has U, right, instead of T. But it's complementary to the DNA sequence here, right? DNA sequence 1 is complementary to RNA sequence 1, right? Except this is DNA and this is RNA. Do you all agree? Do you all see that? All right. It's important that you see that because the next step is we're going to change it. We're going to change it. We're going to cut pieces out. All right? This is not complete. This is too long. There's stuff in this. So this is the gene. That's what's in the DNA. But then your body decides, via evolution, for whatever reason, and we're not going to get into it. There's all kinds of books written on it. There are sections that are removed. This section we take out. This section we take out. We call these post-transcriptional, or these are changes to the mRNA after the mRNA's been transcribed. Post-transcriptional modifications, right? So we have these other enzymes. I'm not going to tell you what they are because you don't want to know. But there are enzymes that take sections off, and we take out what we call introns. The introns come out,
And the extra.
and the exons are left.
Yeah.
These are exons.
Oh, exons. I thought it was extrons.
Yeah, I like that. I think extra-ons are probably better than exons. I like that. But these aren't the extra, though. And I don't even like extra. I almost always think of these as reversed in my mind. Like, I think
Right.
introns are the ones you should keep in, and exons are the ones you're taking out.
Yeah.
But in my mind, my mind doesn't matter. It's what it is, right?
It's reversed.
And then that's right. And then something called the spliceosome sticks them together.
Splicing. What?
It splices them. Splicing means sticking them together.
Say it again. Splices. Splices.
Spliced by an enzyme. Splice means they're you got these three pieces. You take out the introns. Now you got these three kind of fragments. you stick them together, like cutting pieces out of a strip
Oh,
and
it's
taping
sealing
them together. It's an enzyme that sticks it together, it's the tape that sticks together mRNA. And then it goes out to the protein. So this is transcription, this is all there is to it. Tomorrow we'll do translation, and you do an activity where you're actually gonna do the translation. So you're gonna make a protein tomorrow on paper in a lab. So make sure you're, hey,